Okay, we're going to get going. So we had a switch in the schedule, and C.Harris is going to go second. E.Szabo is going to go first. So Eva's chief of the Lung and Upper Aerodigestive Cancer Research Group in the Division of Cancer Prevention. She graduated from Yale University, got her medical degree at Duke University, did an internal medicine residency at Bellevue, New York University Medical Center, and then she came to NCI, her title, non-small cell lung cancer. Eva.
All right. Of course, I have no pocket. Okay, we'll put this. Okay, can here. Can everybody hear me? Okay, very good. So this is really strange because I see 5 of you out there, but I know that there are others elsewhere. So online, okay. So anyway, we'll go forward. So Terry asked me to talk about non-small cell lung cancer. This is a very broad talk. It covers everything from treatment to prevention, which is my area, to early detection. So bear with me as I go through all of this. So I've been doing this talk now, I don't know, 10 years, whatever, more, long time, from the start, whenever that was. And here's the number. Every year, as the population grows, we still get more new cases of lung cancer. It's not really, although numbers per 100,000 may be not going up as rapidly, the absolute numbers are still going up, as are the deaths. Lung cancer is a major cause of morbidity and mortality, more so than anything. It's the leading cause of cancer death in America. It's a huge cause of mortality worldwide, more than breast, prostate, and colon cancer deaths put together in America. And although deaths per 100,000 are decreasing, the absolute numbers are going up and up. Okay. Survival, on the other hand, while it has improved in the past 50, 60 years, it hasn't improved that much. It's still 16% 5-year survival. And that's because you don't usually find lung cancer early. And we'll get into that a little bit. So here's, this is from American Cancer Society publications and website. After people started smoking, 30, 40, 50 years later, you see lung cancer peaked. And as the rates of smoking have gone down, lung cancer is coming down. This is men. Of course, in women, women started smoking later. And so the peak occurred later. It was more in the 1990s, 2000s. And it is starting to come down, but, you know, it's still a major cause of problems in the healthcare universe. So this is one of my favorite pictures. It's an old CT scan. Okay, here's the spine, here's the heart, these are the lungs, and this shows you what causes lung cancer. So this little thing is cancer itself. Here you have some fibrosis, scarring, COPD, okay, chronic obstructive lung pulmonary disease or emphysema, and over here you have the pack of cigarettes. And there you have it, the main causes of lung cancer. Okay, so risk factors, about 85% of lung cancer is due to tobacco exposure, and that includes passive tobacco exposure, although obviously active is much more important. Prior aerodigestive malignancies, most of which are associated with smoking, are again, a risk factor, as is emphysema, chronic obstructive pulmonary disease. Then there's a whole slew of exposures. One that most people know, of course, is asbestos. Think about asbestos exposure and mesothelioma, right? That's what you find on TV. But in fact, the main cancer that is associated with lung cancer, with asbestos exposure, is actually good old lung cancer. Radon, various industrial exposures, oil refining, shipbuilding, et cetera, mining, arsenic, et cetera. There are genetic predispositions. The what's been most notable in the literature recently are these SNPs in the acetylcholine receptor on chromosome 15q. And what's interesting and helps to tie things together is that the same abnormalities or the same SNPs are actually a susceptibility focus for COPD, putting this together. So you're more likely to smoke if you have certain versions of the genes, and you're more likely to get lung cancer and COPD, putting it all together. So we're going to start very globally in terms of clinical presentation, what you do with people, how you treat them, and then move forward, so to speak, for the treatment and prevention. So, yes, ma'am. Even closer? Okay. Is that better? It is. Okay. Sounds the same to me. Okay. I'll go like this. That sounds better. Okay. So we treat lung cancer very much in an anatomic fashion. If it's small, early stage, 1 or 2, small size, T1, the survival is better. The moment it starts to spread to regional lymph nodes, so now we're talking stage 2, wherever we are, okay, stage 2, then the survival plummets into the 30s, 5-year overall survival. And once you get to the mediastinal lymph nodes or to systemic disease, long-term survival is extremely poor. Pathology, okay? The most common form of lung cancer is actually adenocarcinoma, about 40% of all of lung cancer. It tends to present peripherally. These are just photomicrographs. The next most common is squamous cell carcinoma. It used to be the most common, but as the type of as cigarettes have changed and how people smoke has changed, adeno-CA has replaced squamous. Both are associated with tobacco exposure, although lung cancer in never smokers tends to be adenocarcinoma and much less rarely squamous. Large cell is much less common. There's now molecular evidence that really large cell belongs into other categories, such as adeno, squamous, or small cell. And a very small percentage of cases of non-small cell are carcinoids, which are much better behaved, much less genetically abnormal. Small cell lung cancer is the other large category that is associated with tobacco exposure, about 20% of all cases. It's a systemic disease. I think you had a different lecture on this, so I'm really not going to discuss it here. So today, it's really all about non-small cell lung cancer. So if we're going to tackle this big healthcare problem, lung cancer, how are we going to do it? We can do it through prevention. We can do it through detecting it early, stage 1, for instance, when it's much more curable, or we can figure out how to treat it. Ideally, you treat, because then you know who's got the disease. You don't need to bring a larger group into the healthcare system. But treatment, as I'm going to show you, although a lot of work has been done recently, it still has major limitations. And the whole process of lung carcinogenesis is a long process. It starts with tobacco exposure, which for most people starts in their teens. But people don't get lung cancer in their teens. They get it in their 40s, 50s, 60s, the older you get. So it's really this long lag time when you get a series of accumulating abnormalities, both histologically, what we talk about as metaplasia, dysplasia of increasing grades, and molecularly. So if you can intervene during this early process, before you get an overt cancer that crosses through the basement membrane to invade, you potentially could do much better than treating an already metastatic cancer. So, of course, we'll start with the treatment, nevertheless, where you can't cure people. So treatment is very much based on the stage. Early stage, surgery. Early stage with lymph nodes, surgery plus a little bit more adjuvant chemo, it does better. You get about a 5 to 10% absolute decreased risk of death, but nevertheless, the long-term survival is not that great. Once you get to regional spread, then you're talking about combined modality therapy, usually chemotherapy and radiation, because you want systemic exposures as well as local extirpation of the tumor, sometimes with surgery. And then once you get past that, where the cells have escaped from that regional central focus, then you're really talking about chemotherapy and the use of radiation as necessary for local control. Occasionally, you get resection of isolated metastases, such as in the brain. That's basically non-small cell. For small cell, it's all about chemo plus or minus radiation. Very little surgery. What's really become exciting in the past few years, however, is our understanding that non-small cell, which used to, when I was a fellow, I don't want to say how many years ago, it was non-small cell versus small cell. And the definition was histologic, but really because small cell was chemoresponsive and non-small cell was not. And now we have finally learned that not all non-small cell is the same. In fact, these various genetic subgroups act different, behave differently, and we finally have some tools for treating them differently. And that's actually been incredibly exciting and incredibly fulfilling as a clinician. So the one that perhaps you've heard about is the EGFR signaling pathway. It's a very important growth factor pathway involved in many normal processes. All cells have EGFR and molecules that bind to it to set into motion a number of different survival and proliferative pathways. But what's really become very exciting is understanding that there are certain inhibitors, small tyrosine kinase inhibitors, small molecules, which have very profound effects on a certain percentage of people with non-small cell, namely about 10%, and it's the people who tend to be never smokers, often female, often Asian, but who have EGFR mutations, and a very discrete series of mutations in exons 18 through 21. And these people respond beautifully, usually, to EGFR inhibitors, although these responses are short-term. So at the current time, and by short-term, I mean more like 14 months, 8 to 14 months, as opposed to chemo, where the responses are 3 to 6 months. So much better than chemo, but still not cures. So there are there are 3 main EGFR inhibitors that have been developed. Erlotinib is actually the main one on the market, but it's not the only one in the US. Afatinib was also recently approved. And these drugs are now approved for first-line, second-line, third-line treatment of stage 4 non-small cell lung cancer. This is just one study that shows these responses. So typically in all comers, the median survival and deaths is about 10 to 12 months for people with stage 4 lung cancer. This is a study showing in people who had EGFR mutations who were treated either with chemotherapy or, in this case, gefitinib, Iressa, the first of these drugs to go on the market. And what you see is that the progression-free survival, PFS, in other words, the time between diagnosis and progression, according to CAT scan, is very significantly increased. Instead of 5 months with chemo, it's more like 10 months. You see it here, okay? And overall survival in these people is on the order of 30, 36 months, okay? In this study, there's crossover, so people who didn't start out with gefitinib were allowed to get it after they progressed. So you don't see a difference in median survival, but these numbers are substantially better than people who don't have EGFR mutations. So this has really revolutionized how we look at people with lung cancer. First thing, if you have an adenocarcinoma, first thing you do is you look for EGFR mutations, and if you find them, the sensitizing mutations, then that's how you treat. This is EML4 ALK fusion gene. It is another abnormality that is found in non-small cell lung cancer. Again, mainly in never smokers, much less common than EGFR mutations, in somewhat less than 5% of all comers. Although there is a description of a distinct histology, these signet cells, they look like rings, nevertheless, anybody could have it. And once again, there's a very specific inhibitor, and now second-line inhibitors, crizotinib in this case, where once again you have a 60% response rate and a lot of stable disease, and people have much longer survival treated with this drug than with chemotherapy alone. So EML4 ALK, EGFR are frontline tests that we do on all people with stage 4 non-small cell adenocarcinoma, and if they have the abnormalities, that's what they get before chemotherapy. Rest of the mutations are much, much less common. I like to present this case because this is somebody who we saw here at NCI, and this is a gentleman, again, a never smoker, who had a HER2 amplification. So in other words, a different part of the EGF family, EGF receptor family. This is a mutation. This is a abnormality that's found in breast cancer primarily. But in this case, this gentleman was on one of our studies where he had been treated initially with chemotherapy, had very transient response, was treated with erlotinib, Tarceva, so the main EGFR inhibitor on the market, did not respond at all, and then went to a experimental drug, which is not only an EGFR inhibitor, but a HER2 inhibitor. And he actually had quite a nice response, which you see right here. This is his tumor, one of many. And this is what happened after he had this drug that was a HER2 and EGFR inhibitor. But it was, again, a transient response. But what was noted is that his serum HER2 levels were elevated. So we went and looked at his tumor, looked to see whether he had whether he had HER2 amplification. In fact, he did. So we started him on Herceptin, drug that is used for breast cancer, HER2 amplified breast cancer. And actually, he had no response. But when we added vinorelbine, which is a standard chemo agent, which usually has a duration of response of about 3 months, okay, he had a complete response for 13 months, had one little thing that grew out in his neck. We took it out. We put him back on these drugs. He had another lengthy response. And this gentleman has now been alive for 7 years with metastatic lung cancer, going from one HER2 targeted therapy to another. Actually, an outlier, without a question, but a remarkable case. And this is what we're up to now in lung cancer. This is ROS1, again, a tyrosine kinase rearrangement that occurs in a very small number of non-small cell lung cancers, but these people also respond to crizotinib. So we're starting to whittle away at these small subgroups of people, which has its own challenges because you have to look for these abnormalities molecularly. But when you find them, the responses are dramatic, they're much longer lasting than chemo, and they're very satisfying, obviously, to the patient, but even to the physician. Okay. But that's really adenocarcinoma, the majority of lung cancer. What about squamous cell? TCGA and the Cancer Genome Atlas project, as well as number of sequencing studies, have identified certain abnormalities, such as the fibroblast growth factor receptor 1 that are abnormal. This DDR2 is another tyrosine kinase mutations occur in squamous cell carcinomas, and there are experimental drugs that are effective against these two. So we don't have quite the level of evidence, but at least we have a place to go with some of these abnormalities in terms of clinical trials. So that's really the status of the genetic approaches to non-small cell lung cancer. What's perhaps in its infancy right now, and it's not an area that I can speak to very well, but what is really exciting is immunotherapy for non-small cell. And in particular, the checkpoint inhibitors, this PD-1 molecule, which is involved in T-cell activation, actually in limiting anti-tumor immunity, and inhibitors, which are primarily are antibodies, have shown remarkable efficacy. And when I say remarkable, if you look at the response rates, which are on the order of 10 to 25% response rates, perhaps that's not as exciting on the face of it, but what's really exciting is the duration of these responses. So on this one trial, which uses a Merck compound, which is an anti-PD-1 antibody, this response rate yielded a median survival of 51 weeks, which is a year, okay? But what's really remarkable is that for the people who respond, the majority respond long-term, and not necessarily even while they're still on drug. So they're people who are years out from this kind of therapy. In other words, there's a tail to the survival curve, and that's a first for non-small cell lung cancer, because even with EGFR inhibitors for EGFR mutated non-small cell, there is no tail. People die. That tail goes to zero. So this is really the forefront, and hopefully this will make a difference, although again, it appears to be in a minority of people, and mainly in people whose tumor expresses the ligand for this PD-1 receptor, namely PD-L1. Okay, that's it for treatment. It's an overview. It doesn't do its justice. I apologize, but at least you heard the buzzwords. And those are EGFR, EML4 ALK, PD-1, so on. The rest of the time, I'm going to be talking about other approaches to limiting morbidity and mortality from lung cancer, namely prevention, and a little bit on early detection. So again, in the sequence of events, the early phases of lung carcinogenesis are before the abnormal cells develop enough abnormalities that cause them to go through the basement membrane and out into the bloodstream or the lymphatics. Can we do something to prevent that process from occurring? Well, let's start at the root cause, okay, smoking. What happens if you stop smoking? Well, it's not as simple as one would seem. If you stop smoking, 5 years later, your risk of lung cancer hasn't perceptibly gone down. This study here, which is the lung health study, looked at 14.5-year follow-up, median follow-up, in a group of people who either intermittently quit, or continued to smoke. And what you see are the causes of death 15 years later. And what you see is that for the people who quit, you basically have the number of deaths from lung cancer. People who quit intermittently fall in between, and people who continue to smoke are the ones who have the highest mortality from lung cancer. Again, when in the same study, looking at the same kind of differences in survival due to various diseases, the 5-year follow-up did not show any differences in lung cancer. So it really takes a long time before you start to see a difference. The take-home is not that you shouldn't quit smoking. The take-home is that you're not going to see the benefits for lung cancer until years down the line, whereas you see them quite quickly for cardiovascular disease, your ability to breathe better, non-progression of emphysema, and things like that. But quitting smoking is as hard as everything else. It sounds easy, it's not. And so the current adult smoking rates are on the order of 22, 23%, very high, okay? So is there something else we can do? And that's where chemoprevention comes in. So chemoprevention is defined as the use of strategies, natural, synthetic, immune strategies to suppress or reverse the process. Here the emphasis is not on killing certain cells, it's on reversing a process that occurs over a long period of time. So regressing the existing preneoplastic lesions, preventing the development of new ones, suppressing the recurrence of previously treated overt cancers. And the rationale comes from a variety of reasons or sources. And it really starts with the fact that if we could treat metastatic cancer, we wouldn't want to focus on those early timeframes because not everybody who smokes goes on to develop lung cancer. So we'd want to focus on those people who have cancer already, except that we can't cure metastatic cancer. We can extend survival, but even with the best therapies that I just showed you, we don't do that well. People aren't cured. And we do have data from other diseases that cancer is preventable, in fact. In breast cancer in particular, using tamoxifen or raloxifene or aromatase inhibitors, one can prevent second primaries, contralateral primaries, or first cancers in people who are at high risk. We can model this in animals with carcinogen exposure, various genetic abnormalities. And we also know that it takes a long time to develop all those abnormalities that eventually lead to invasive lung cancer. So we've got a population at risk. We have a long time frame for intervention, and we know that it can be done, at least, it can be modeled in other diseases or preclinically. So what do we want to do? When do we want to intervene? So again, if you have an effective intervention late stage, you don't need to go to the early stage. But early stage cancer, by and large, is much more curable than late stage. Stage 1, of course, has much better survival than stage 4. One would hypothesize that precursors would be more curable than invasive cancer. They're not as genetically complex. Could we prevent carcinogen damage? Yes, but then you need to start with your teenagers. But that would be the ideal thing. These are all issues. It tells you when you want to focus. The other side of the equation is the toxicity. You can give highly toxic agents in the short term, for instance, when somebody has metastatic cancer, but you can't do that in the long term. Long term, you need to go with very non-toxic therapies. So that is obviously a limitation and an important one. And you also have to think about things in terms of public health. The target population to treat is huge, right? If you take all the current and former smokers, that's over 90 million people in the United States at any one time. So obviously we can't treat everybody. We don't want to treat everybody, but we certainly can't afford to treat everybody. So these are all issues that one needs to think about when contemplating how to bring prevention to large population. It's all about the risk and the benefit. Benefit, the efficacy in preventing cancer and its associated morbidity and mortality. Risk in terms of the side effects, the adverse events that increase morbidity and mortality from other diseases. So in other words, you prevent the lung cancer, but if people die from heart attacks, you're not really doing anything good, right? And also the tolerability. You can give people side effects that we don't consider medically important, but if a person won't take the drugs because they're having diarrhea, let's say, it's not going to work. So all of these things go into the risk balance. Okay, so again, just a little bit of background. How do we identify new agents for lung cancer? Mechanism, where we know it. HPV vaccine is the best example of mechanism. HPV, human papillomavirus, is absolutely required for cervical carcinogenesis. Vaccine prevents it. Beautifully modeled in animals and now even in humans. So when you know the molecular pathogenesis, that's great. Lung cancer is a little bit more complicated than that. So we often go to preclinical models, animal models, as well as in vitro systems. And I will show you examples of ones that have led us to clinical trials. We go to observational epidemiology, cohort and case control studies, which show us what agents may be of potential utility. I'll show you examples of that. And we also look at secondary endpoints from clinical trials that were perhaps performed for other diseases. Best example of drugs coming into cancer prevention via this route are with tamoxifen, where tamoxifen was used for adjuvant treatment of breast cancer, and it was found to prevent second primaries, et cetera, et cetera. And I'll even show you examples of where we're going with that with lung cancer. So I'm going to tell you 3 different stories, short stories, of areas that we've been focusing on, okay, for lung cancer prevention. One of them is the inflammation story. Long history, okay, animal data going back 40 years or more, showing that corticosteroids, which are anti-inflammatories, can prevent skin cancer in animals. That work was extended to look at lung cancer models, again in animals, using oral and inhaled steroids. And there's even some epidemiological data, which really is reasonably new, showing that in people who have emphysema, COPD, who are treated with corticosteroids as opposed to other agents, that the incidence of lung cancer is much decreased, okay, hazard ratio of 0.39. So animal data, epidemiological data. Let me show you a little bit of the animal data. This is one example using a carcinogen called vinyl carbamate, which produces adenomas and actually adenocarcinomas in animals that are treated with vinyl carbamate. If they are given budesonide, which is a steroid, you get an 80% decrease in the number of tumors that these animals have. And if you look at what these tumors look like histologically, they are more likely to be adenomas and less likely to be carcinomas than in the untreated animals. These are untreated, these are treated. So you have the numbers of carcinomas. So what you're doing is you're reducing the total number and you're preventing the progression. So based on these data, we worked with Stephen Lamb and the British Columbia Cancer Agency to do a bronchial dysplasia trial. This is how you do lung cancer trials. So we took about 1,000 people who were current or former smokers, looked at their sputum to see whether there's any atypia, identified about half of them having atypia, asked them to have a bronchoscopy to look inside the bronchial airways, and identified finally 112 people who went on study who had bronchial dysplasia and who had 6 months of either an inhaled corticosteroid, budesonide, which is used for approved for asthma, or a placebo. And then they underwent a second bronchoscopy. They also had helical CTs to look at their peripheral lung, okay, to see whether they have any nodules. This was, again, based on the animal studies and everything that I just showed you. So what we found was that actually there was no effect, a very disappointing, large effort, phase 2 study. What you see is that about a third of the people undergo spontaneous regression of these dysplasias. There was no difference in the progression rate, in other words, new areas of dysplasia. But when we looked at the CT-detected lung nodules, there was a statistically significant decrease in the number of nodules. So that gave rise to the second study, and this is the first of its kind, where we looked at people who had CT-detected lung nodules. These folks were undergoing CT screening. They were part of a screening trial at the European Institute of Oncology in Italy. Giulia Veronesi was the PI, large program there. And we took 200 people, 202, who had nodules and treated them either with inhaled budesonide or placebo for a year, and then asked what happens to the nodules. Okay. So the primary endpoint was just nodule size, no histology, because these are small nodules. And there, what we found is that in the non-solid nodules, the ground glass opacities, there was a difference. Budesonide made people were much more likely to regress, have smaller nodules or disappearance of nodules if they had the inhaled steroid. But overall, there was no difference. And overall, there was no difference because the majority of nodules are little tiny solid nodules that simply don't change in size at all. Now, we think that it's the non-solid nodules that are the important ones. There's accumulating data in the literature that a proportion of these non-solid nodules, you see one right there, this is what it can look like under the microscope, a reasonable proportion, quarter to a half or more, are either early bronchoalveolar carcinomas or atypical adenomatous hyperplasias, which are precursors to lung cancer. So the new studies are all going to be looking at only these types of nodules. We didn't think we had the power to look in the budesonide study because we didn't have enough people with these small nodules. So the inflammation story is not quite over. You know, the 2 trials that we did were not positive, but they show us where to go. And they may not be positive because, indeed, it may be the wrong hypothesis or the hypothesis is incorrect. It may be that we're simply not getting enough of the inhaled steroid peripherally enough to get an effect. There are a number of logistical issues. What we still don't know is which are the best nodules to go after, but we think that they are these ground glass opacities. We need to learn more about them. We need to follow these people long term and to do studies with perhaps more effective agents. So is there a more effective agent? Maybe. This is aspirin. We go back to where everything starts. So this is from a series of studies by Peter Rothwell in the UK, looking at long-term follow-up of people who were treated with aspirin or not, or placebo, mainly for cardiovascular reasons. And what you see is that after 5 years, the 2 curves splay. There's about a 30% decrease in lung cancer death, not just incidence, but death after 5 years of aspirin treatment. It's actually only adenocarcinomas. It's not squamous cell carcinomas. But this tells us that if, and again, these were intervention trials. This is not plain epi. This is a meta-analysis of a series of intervention trials. So this tells us that if you're dying at 5 years, your incidence, your diagnosis would be 3-4 years prior to that. Your stage 1 lung cancer would probably be 4 years prior to that. Your pre-malignancy would be even earlier. So this gives us a window for doing studies with aspirin, looking at those ground glass opacities, and that's exactly where we're going right now. Let me tell you a second story. This is a drug called Myonositol. Okay, it is actually a food constituent. It's a glucose isomer. It's actually a source of several secondary messengers and signaling molecules, and it's relatively benign. It's something that we call GRAS by FDA technology, where that's a designation that is generally regarded as safe. It means you don't even have to do the paperwork for an IND to use this drug in studies. Based on the work of Lee Wattenberg and others, there's a lot of preclinical data for this drug alone or in combination with inhaled steroids. In any case, going back to Stephen Lamb, we did a phase 1 study to look at what's the best dose. And here, in this phase 1 study, what Stephen found was that the regression rate of dysplasia after 2 months of Myonositol was striking, 91% compared to historical controls. Very important. These are historical controlled. This is not a controlled study. Interestingly enough, there was also a slight decrease in blood pressure. That was not to abnormal levels, but from high normal or just slightly supernormal to more normal levels. So it's a sort of an exciting concept that perhaps this would be a drug that would also have other good effects. Now, what Stephen did, which is quite novel, is that he also did bronchial brushings from the normal epithelium. So he looked at all the areas of dysplasia, and then he brushed the normal bronchi and sent the brushings to Avi Spira at Boston University, who did gene expression analysis. And what Avi found, looking at these samples and many others, is that people with bronchial dysplasia have upregulation of PI3 kinase signaling. And the people who responded by reversing their dysplasia in this small cohort also reversed the PI3 kinase signaling. So now we have a potential molecular signature that we can look at, both as the people at risk, they have PI3 kinase activation and dysplasia, and as a readout, as an endpoint, those who reverse one reverse the other. That's the hypothesis. So we now have potentially a way of moving forward, which is really a new clinical trial model, which we will be putting into place. So, questions. Does PI3 kinase activation truly identify the at-risk smokers? Because they're the ones who have the dysplasia. Okay. It would certainly be easier to get those brushings than to be looking for dysplasia. And there's data that you get similar changes in the bronchi as you do in the nasal epithelium. So this is all an evolving area. But it also gives us a new clinical trials model where we look at the pathway analysis pre and post treatment. And in fact, we're looking at that with aspirin, no less. So there is an ongoing study which is duplicating this, but in a much larger number, using Myonositol, the same drug, looking at dysplasia, but also doing the pathway analysis from the normal bronchial epithelium. And this study has 85 people on it, and we should have the results before I give this lecture next year. Okay, one last area to talk about in terms of prevention, which is the Peroxisome proliferator activator receptor gamma, PPAR gamma, which is another potential target that we've been looking at. So there are drugs on the market for treating type 2 diabetes. Pioglitazone is the main one in use today, approved for a number of years. And in data and work that really started with cell line studies and then progressed to animal studies, we were able to show that treatment of lung cancer cell lines induces growth arrest, differentiation in vitro. There are animal models. This slide I show you an oral, a tongue model, where you have much decreased incidence of tumors, and again, decrease in carcinomas. There's some epidemiological data, about a 33% decrease in lung cancer incidence in diabetics who use this group of agents compared to those who don't, and even higher decrease in head and neck cancer, all part of that aerodigestive tract. So the way we moved forward with this drug was to do a small phase 2 study. And actually, we didn't do it in lung cancer. We did it in oral leukoplakia. Oral leukoplakia is a precursor to oral cancer, but again, it's part of that aerodigestive exposed field. And in this small study, 22 patients, we found an 80% clinical response rate with a huge decrease in size of these lesions. A little bit hard to see, but there is this whitish plaque on the tongue, which is not there after treatment. So the current ongoing study is, again, ramping this up, double-blind, placebo-controlled. There's data that, and this is all head and neck cancer, but there's data that you have similar efficacy in lung cancer. There are preclinical studies, again, looking at mice with and without p53 mutations, huge decrease in tumor burden in a preventive setting. And so we are only doing the oral leukoplakia study, and we did a small pilot pre-surgical lung cancer trial, but there is an ongoing study by the group in Colorado that's looking at pioglitazone in people with bronchial dysplasia. So in the next couple of years, we'll have data. Okay, I'm going to finish up with just a little bit on early detection, because that's actually an area that has really blossomed in the last couple of years. But before I talk about the positives, let me tell you the negatives, okay? And the negatives are that screening is not a freebie, okay? Sounds easy, it's not. And it's because there are these various biases that are well known to be part of screening. Lead time bias, where you diagnose the disease earlier, but you don't actually postpone death. You just know that the person has the disease earlier. Length bias, where you're more likely to diagnose the indolent disease than the rapidly progressive disease that actually kills the person. So you're looking only at the better prognosis, better outcome disease. And of course, overdiagnosis, where you're going to find lesions that are clinically unimportant that would not be diagnosed otherwise. And there's now data that actually all of this occurs in lung cancer too. The best overdiagnosis that we can think of is, of course, prostate cancer, where if you do autopsies on men who are 70, the majority are going to have prostate cancer. Very few of them die from prostate cancer. Not to say it's not an important disease, but it's very common, a lot of overdiagnosis. So early detection has been a big focus, actually, of the NCI, but others for a long time. And chest X-ray was studied, sputum cytology were studied many years ago, 30, 40 years ago, with no good results. Really, the definitive test, definitive screening trial was performed fairly recently, the PLCO, prostate, lung, colorectal, and ovarian cancer screening trial, 2011, showing, I think once and for all, that chest X-ray does not detect lung cancer effectively. What does? Helical CT. So this is the National Lung Screening Trial, which is a very large study, 53,000 smokers, current and former, specific criteria, 30-pack year, quit less than 15 years ago, age 55 to 74. People were randomized to either getting a helical CT, spiral CT, okay, or chest X-ray 3 times. And the results were a very large number of positive tests, okay, but a 20% reduction in lung cancer mortality, and even a reduction, statistically significant, 6.7% reduction in all-cause mortality. So this is the first time that we have the ability to actually prevent lung cancer death, okay? Here's just the slides to show it, or the graphs to show it. So very exciting results. How you get that to 90 million current and former smokers is a challenge. So how do we move forward? A lot of modeling data. There'll be more data coming from European studies. But here is one beautiful example, looking at when you have a nodule at the first screening CT, what's the chance? Who are the people who are really going to develop lung cancer? So this is, again, from the British Columbia group, who looked at a large number of people in various data sets and developed a risk calculator, which one can find at this website. And based on things such as age, sex, family history, emphysema, okay, by history, and then the nodule size, type, location, there's a very accurate way to predict who's going to develop cancer in the next 3 years. And so this is one potential way to figure out who really needs to be screened repeatedly, who should go into chemoprevention trials. So all these things are coming together. We are now at a point where we can actually identify people who have early lung cancer, identify those who need subsequent screens if they don't have lung cancer. I mean, obviously, more validation is still needed, but this is a pretty validated study right here. So a way to move forward, looking at the various pathways, some of which I described, looking at the PI3 kinase signature, et cetera. And so I think we're really now poised to move forward, okay? So in summary, a lot of progress has been made, not just in understanding the molecular biology of lung cancer, but actually in taking it to individual patients, both in terms of treatment, precision medicine, as it's typically called, with clearly increased survival in advanced stage patients, but not cure. Immunotherapy, we're just at the beginning of that whole area, and I think that's going to make a huge difference. Screening with CT, which is going to be refined further to figure out who needs to be screened and how long they need to be screened. And with these new tools and targets, we can now really do chemoprevention studies moving forward. So that's it. Thank you for your attention. Any questions, I'm happy to answer. Lung cancer? It's not. It's actually a very commonly dysregulated pathway. There are actually a fair number of mutations in the squamous cancers. So head and neck cancer frequently have PI3 kinase mutations or PTEN abnormalities, esophageal. And bronchial dysplasia is a squamous precursor. But it's not limited to those either. So, yes, there are some mouse studies which I want to say are still ongoing. Our group is doing that. If you buy that Myonositol is a PI3 kinase inhibitor, which actually in vitro it does appear to be, there are a number of groups. I know there are. And there's a head and neck group that studies mTOR, PI3K, AKT, mTOR. There are a number of groups. So the answer is yes. All right.
Thanks, Eva. Okay, and our next speaker is C.Harris. He's chief of the Laboratory of Human Carcinogenesis. He got his MD from Kansas University School of Medicine. Then he did his clinical training in internal medicine at UCLA. He's received numerous awards, one of which is the NCI Outstanding Mentor Award. And his talk today, Interweaving the Threads of the P53, microRNA, and inflammation networks into the tapestry of cancer and aging.
Is that what I'm going to talk about?
The world traveler is here.
That's thick. Actually, what Terry is talking about, I got back last night from China. So it's sometime early in the morning for me. But Naomi came over to keep me awake and yell at me every once in a while. Handouts, if you don't have one, they've been handed out or they're outside. All right, this thing works, right? Great. All right, I'm going to break this talk up into 4 parts. First is going to be about aging and senescence, and that senescence is a barrier, a tumor suppressor barrier to cancer. The second will be a very short presentation about stem cells and why we're interested in lung cancer stem cells. And then we'll get into more practical stuff about screening microRNAs and their role in prognosis and response to therapy. And there will be some inflammation in there too in regards to risk of disease and the combination of inflammatory biomarkers and microRNAs or protein coding genes as biomarkers of prognosis. So very briefly, cellular senescence is caused by several different mechanisms. First is telomere shortening that occurs during replication of cells. That's the traditional way, and that leads to a DNA damage response that can be measured by gamma H2AX. And in this case, you can see the telomere ends, and the response is at a particular telomere in the chromosomes. And there's evidence that actually DNA repair occurs more slowly at telomeres than does the rest of the genome. It goes through DNA damage signaling cascade and primarily p53, leading to senescence. There are many markers of senescence, including senescence-associated beta-gal. It's a blueness that you can see here, just a change in the pH. A second kind of senescence is just DNA damage that occur at any place on the chromosome or the DNA. And this also goes through p53. And the third is oncogene overexpression, such as KRAS, and this goes primarily through an RB pathway. So these are the people in the lab who've been studying this, and they've been investigating two different hypotheses. First, p53 isoforms, which I'll define, are physiological regulators of replicative cellular senescence, both in vitro and in all of us sitting here in this room. The second is that these isoforms are deregulated during carcinogenesis, particularly lung carcinogenesis and colon carcinogenesis, and also in tumor-associated T cells. So about 90% of genes have isoforms. p53 actually has 13, and their function has not been well studied, but two of these have been investigated by us and a few others. One is p53 beta, which has an alternately spliced carboxy terminus, and the second is delta 133, in which a third ATG site is used as a start site, and so the first 133 amino acids are missing, and it acts as a dominant negative. This one acts primarily as a co-trans activator of p53. So the initial kind of experiment was just to take the traditional fibroblast experiment, grow the cells, young cells, ones that are rapidly growing versus those that stop growing and are senescent, and just ask the question, what's the level of expression of these isoforms? The p53 beta appears only in the senescent cells, and the delta 133 is detectable only in the young growing cells. And these cells already have markers of senescence, including activation of p53 and high levels of p21, which is downstream from p53. The physiological way of, or the best way, in my opinion, of asking the question whether or not a gene has a function that's relevant is to knock it down or knock it out. So we use two different siRNAs here, and as you can see, there's much less delta 133 here, and this is using a second kind of antibody, and you can barely see it here. When we knock down the delta 133, the dominant negative form binds to p53 and inhibits p53 function. You can already see an increase in p21, and you can see these blue cells that are more fried egg-like, and you could quantitate them. So what happens if you do the opposite experiment, and that's overexpress the co-trans activator form of p53, which enhances p53 function, or the delta 133, the dominant negative. So this is a short-term assay just looking at cell growth. Overexpression of beta compared to the vector control inhibits cell growth and induces senescence, as quantitated here with senescence-associated beta-gal activity. But overexpression of the delta 133 looks like it even enhances growth, has no effect on senescence. And then when you do the experiment of taking cells that are near senescent and only have 2 or 3 or 4 population doublings to go before they become senescent, you overexpress the delta 133 and you get about another 10 or 12 population doublings. Well, that's all cell culture kind of experiments. What happens in us and as we age? I thought about this in terms of what cells to do this experiment with, and finally came up with CD8 positive T cells. And there'd been some previous work with this population of T cells in senescence, in the sense that there's already some evidence that as people age, there's a change in these cells, and they became people would call immunosenescent. And I'll go very briefly through this data-rich slide. And what happens is that CD28, a cell surface co-stimulating protein, goes down with increasing age. And the lack of this, the cells without it, goes up, as one might examine. Telomeres are shorter in the CD28 minus cells, and they have an increase in senescence-associated protein. And another senescent marker is HP1 gamma, that's heteroprotein 1 gamma, and it increases, and the quantitation is here. And if you isolate these cells, they have an increase in IL-8 and IL-6 in CXCR1 and CXCR2, and all of these have been associated with senescence of fibroblasts, and IL-6 and IL-8 actually are growth factors for human lung cancer cells. And in fact, many lung cancers produce IL-6 and IL-8 as autocrine growth factors. So what happens with the levels of the co-trans activator or the dominant negative form of P53? So the dominant negative form of P53 goes down, and the co-trans activator form of P53 goes up in the cells that are immunosenescent. We did the similar kinds of experiments with cultured cells from lymphocytes, that is, lymphocytes from people, and took young growing CD8 positive cells and put them in culture, and with time they senesce, they lose their CD8-28 positivity and become negative, and they have an increase in beta-gal activity and HPO1 gamma. Those are again senescent markers, and they have DNA damage, again primarily at telomeres. Even though, and then lastly, these are just the Western analysis showing that the P53, delta 133 P53 goes down, the cells growing in culture and becoming senescence, and the P53 beta goes up, as you can see here. So you could do the other kind of experiment, and that's to overexpress delta 133. Actually, this is a knockdown of delta 133. The cells don't grow, and they become have a senescent phenotype. And or you can overexpress P53 beta, and the cells do grow. Excuse me. They go into senescence. Get that correct here in a moment. So let's summarize that, then I'll tell you how this relates more to cancer. So P53 isoforms, there were 13 different ones. We've identified the RNA splicing protein for the P53 beta. The delta 133 is controlled primarily by autophagy, and it goes down in senescent cells, either fibroblasts or lymphocytes in vivo or in vitro, and the P53 beta goes up. In benign tumors, that same thing happens. Most benign tumors have a lot of senescent cells, but when they become carcinomas, you get this switch in the isoforms, and this occurs also in lung cancer. So the dominant negative form goes up, and P53 beta goes down, and T cells associated with tumors that can have an enhancing effect as well as a anti-tumor effect, and the enhancing effect occurs primarily if the cells are producing IL-8 and IL-6 and are senescent. So as I mentioned, precancerous lesions in benign tumors, whether it's mouse or human, have a high percentage of senescent cells. But in the other this is from various people's data, including our own. And in animal models, if you restore p53 at levels that are relatively physiological as opposed to super high levels that you actually don't see in tumors, tumor regression frequently occurs by senescence, not by apoptosis. So there are a number of mechanisms for induction of senescence in benign cells. I listed at least two here. That's a p53 beta and a decrease in the delta 133, the dominant negative form, an increase in p16 that's in the RB pathway, DNA damaging agents, free radicals such as nitric oxide, and of course telomere shortening and uncapping. And there are two barriers, p53 network and the RB network, that are barriers to and can bypass. You can bypass the senescent barrier by decreasing the p53 beta, increasing the delta 133, but more effective than that is a p53 mutation and DNA methylation of p16, and which usually leads to some decrease in expression. Will bypass this mechanism, but more effective than that is a p16 mutation. So let's move on to a topic of cancer stem cells, and particularly lung cancer stem cells. And why, why am I telling you about this? Well, stem cells, normal stem cells, are important in homeostasis, and in which the stem cell divides and one of the two partners is a stem cell and the other will differentiate. In the lung, it would be a mucociliary differentiation. But under certain circumstances, which I'll tell you about in a moment, you can get symmetric differentiation. So a stem cell becomes two differentiated cells. And in cells that are regenerating, and in some cases cancer, you end up with a symmetric cell renewing, so that you end up with two stem cells. And that's what the hypothesis that we're exploring in tumorigenesis, that cancer-related genes, particularly cancer driver genes, push this option towards symmetric cell renewal. Well, you can measure asymmetric cell division and symmetric cell division, and let's first look at asymmetric cell division. And these experiments you use BUDR. You go through two cell replications, and then you isolate anaphase cells. And what one finds is that BUDR is in one of the two halves of the cell. So this is an anaphase cell. It's going to divide right here, and the stem cell is here, and this is will be the differentiated cell. So you can actually measure this in cells in culture, either immortalized non-tumorigenic bronchial epithelial cells, or in various lung cancer cell lines, or in colon cancer cell lines. And cell lines are okay for doing certain manipulations, but they're quite far from the original tumors. And when we looked at primary lung cancers and put the cells in culture, we found that the frequency of asymmetric cell division was quite high compared to the cell lines, most of the cell lines. But with increasing selection in culture, the asymmetric cell division rate went down, and we were proposing, or we do propose, that might be an increase in symmetric self-renewal instead of asymmetric. Now, you can measure stemness by different ways in lung cancer as well as other cancer types, but one cellular marker is CD3133, and you can measure it as shown here, and then when you do the experiment with the BUDR, you can find BUDR and the stemness marker going together. But what's happening to this particular portion, the cell that would be the progenitor cell from the stem cell? Well, you can use markers of differentiation, such as surfactant, pro-surfactant marker. And as you can see here, the pro-surfactant marker is only on the side of the non-stem cell or non-BUDR, and the pro or the pan-cytokeratin is also only on the side of the non-stem cell partner. So does P53 or other agents modify this? P53 comes in many different flavors. These are all somatic mutations, and one can do experiments taking P53 null cells and overexpressing different mutants, and some mutants have been shown to have gain of function, and some have just loss of function. And so we used a DOX-inducible system. As you can see with DOX, there's expression of these various mutant forms of P53, and the degree of asymmetric cell division goes down at least with 2 of these in both cell types here. And if you remove the DOX and the mutant protein goes away, the asymmetric cell division frequency goes up. So that's a good control, saying that it's being modulated by the mutant P53. So how do you measure the other half of it? And that's symmetric self-renewal. And so Wakefield has made a construct called Spore6 that is activated by OCT4 and SOX2. These are stemness transcription factors, and you can measure that by FACS analysis and by fluorescence. And when you isolate cells from cell lines, in this case initially, and isolate the cells that are positive for Spore6, they form tumor spheres in culture where the ones which are negative rarely do. So we're currently testing the hypothesis that mutant P53 increases self-renewing symmetric cell cancer cell division. So Anna Robles and I recently wrote a commentary that appeared just a few days ago, actually, on a paper that was measuring reportedly cancerous stem cell effects or the effects of a primate-specific microRNA on symmetric and asymmetric cell division. So in this cartoon, this is a normal stem cell, and as I mentioned earlier, normal stem cell primarily goes through asymmetric cell division, with one cell being the stem cell, so it persists, and then the other partner becomes, or the progenitor cell becomes differentiated, in this case mucociliary differentiation. But under chronic damage or stress, you can get symmetric differentiation, and this can lead to squamous differentiation. Or under acute damage and stress, you can get symmetric cell self-renewal and regeneration, for example, basal cell hyperplasia. So what is hypothesized is in cancer stem cells that can occur through a progenitor cell, or I think much less frequently arising from the stem cell itself, the asymmetric cell division can occur, and with self-renewal, and symmetric self-renewal, and could be objects of targeted therapy. And we're proposing that this is driven primarily by cancer driver genes, such as ALK fusion genes in lung cancer. And the primate-specific microRNA, in this case, this particular one, is involved in a terminal differentiation. And as an aside, primate-specific microRNAs make up about 1/4 of the total number of microRNAs that have been described so far. And there's over 2,000 now. And so you can make the argument, well, why are they primate specific? And there's even one that appears to be human specific. Now, what's the evolution of that? What are their particular functions? Are they redundant functions, or are there brand new functions that have evolved? Anyway, so it's another area of microRNAs, which now I'll talk about. So microRNAs and cancer, interesting story over the last 2 decades. First of all, Victor and Gary Rufkin made the initial discovery in studying C. elegans. And they found that there were small non-coding RNAs, about 26 base pairs, that were evolutionarily conserved and regulate gene expression. And so they were basic scientists, and their observations actually were not well noticed until about 10 years later, in which microRNAs were found in human cells and tumors, and they were differentially expressed in human cancers. And Carlo Croce did the seminal, made the seminal observations at that point in chronic lymphocytic leukemia, in which MIR15 and MIR16 were deleted. So that was 10 years after these basic research findings, so hooray for basic research, and hooray for using basic research into the clinic. And I will say that microRNAs are evolutionarily conserved again. So you can find microRNAs in plants as well as in humans. So they were only recently discovered, but very ancient mechanism of epigenetic changes. So the rest of the talk will deal primarily with microRNAs and inflammation for predicting risk, diagnosis, prognosis, and therapeutic outcome. So microRNAs are encoded by genes, and Pol2, RNA polymerase 2, encodes most of them. They form initially a prior microRNA, the stem loops kind of structure. It's processed in the nucleus by Dortier or a variety of other proteins in a complex. Dortier is an RNase, and it cleaves part of this, so you get the pre-microRNA. Then it's exported into the cytoplasm. It interacts with a second RNase called dicer, which is part of a complex, to cleave this down to 26 base pairs. And then in another third complex called RISC, it forms a complex with message, protein message RNAs. It has 2 main functions. It can either repress the translation of the message or cause instability of the message. And that's shown here again, translational repression or message cleavage. Another function of microRNAs that's been discovered is that these small microRNAs combine to proteins and inhibit protein function. In this case, an RNP function that is important in granulocyte differentiation. So it's an inhibitor, actually. So this particular microRNA inhibits an inhibitor of protein transcription. And lastly, microRNAs can act as ligands to receptors, including toll receptors, TLR8 in humans and 7 in mice. This activates NF-kappa B pathway, and you get high levels of IL-6 and TNF-alpha. And the third basic aspect of microRNAs is that they can be transported from one cell to another. And this can be through exosomes. So exosomes are budded off of cells, and this is a normal process that can occur from plants all the way to humans. And these exosomes contain various proteins and microRNAs and in some cases message RNAs. And then they go out into the body fluid, and you can isolate them from serum and from plasma but also from lymph, and they fuse with other cells, in this case an immune cell such as a macrophage, and the contents of the exosome are released into the cytoplasm and can activate, in this case, a toll receptor, and you get a feedback loop. So they're very adaptable molecules that have many, many different functions. So we're quite interested in mechanistic biomarkers of cancer risk, prognosis, and therapeutic outcome. For risk, we heard a presentation about that. Prevention and screening are important, and gene-environment interaction. Prognosis, to treat or not to treat. And most stage 1 lung cancers, which I'll talk about, are not treated other than surgery. But 30% of those folks are going to die of recurrence. So they die of micrometastases that are not detected. And then there's interesting biology and development of therapeutic targets. So this begins with a study we did with Carlo Croce 7 years ago now, and we asked a very simple question. Which of the microRNAs are most highly overexpressed in 6 major types of human cancer? Breast, colon, lung, pancreas, prostate, and stomach. At that time, there were only about 280 microRNAs that were known, and Carlo had developed a chip, a microarray for measuring these microRNAs. And one came up to the top, and that was miR-21, and that's what I'll spend most of my time talking about. In all 6 of these, it was highly overexpressed. And now miR-21 is upregulated in 18 major cancer types and a prognostic biomarker in at least 12 cancer types. So this is low-hanging fruit, and that's how we pursued it. There's some interesting mechanistic data I'll tell you about toward the end. This is a picture of Nozumo, who was in the lab at the time, and so he tests the hypothesis that microRNAs are associated with lung cancer diagnosis and prognosis, and he found that microRNAs are significantly different between both primary lung cancers and the corresponding non-cancerous tissue, and between different histological types of lung cancer. And that there was increased miR-21, miR-155, and 106 in these lung cancers, particularly in stage 1 lung cancer, again, the earliest stage of lung cancer that we tend to focus on, that were associated with diagnosis and prognosis. And the LET7 decrease was confirmatory of a study that Takahashi did, and also Frank Slack in lung cancer in regards to diagnosis as opposed to prognosis. So one important aspect of doing these mechanistic and biomarker studies is to look at multiple cohorts in different geographic areas, different ethnicity. So we follow that up with examining 3 different cohorts, one in Maryland, one in Norway, and one in Japan. And these are Kaplan-Meier plots. So 100% survival, 0% survival, this is the 5-year point. So high levels of miR-21, above the median, poor prognosis in each of these cohorts. Now, has this been found in other cohorts? The answer is yes. We discovered this also in lung cancer cohorts in 1, 2, 3, 4 different cohorts, and in 3 different cohorts that I just mentioned, and colon cancer in 4 different cohorts. And these are studies done by other people, and almost all of these have been replicated by others. And and there are only 10 out of the 12 or 13 that I've shown on this particular slide. So high levels of miR-21 are associated with poor prognosis. Now, can we combine microRNAs with inflammation? And inflammation, as you've heard, is associated with increased risk of cancer. It can be either inherited, such as hemochromatosis, and iron overload disease with liver cancer, or inflammatory bowel disease or familiar pancreatitis. But more frequently, inflammation is acquired either due to viruses or bacteria or parasitic infections. And it's been now recognized that chemical, physical, and metabolic conditions lead to inflammation, including acid reflux and Barrett's esophagus, increasing esophageal cancer. Asbestos does many things. It actually damages DNA, but it's also an inflammatory agent. Obesity is a chronic inflammatory disease. And smoking, there's, I think at last count, 62 chemical carcinogens, known chemical carcinogens found in tobacco smoke. But, so it's a witch's brew of carcinogens. But any of you who have ever taken your first puff of a cigarette realize how toxic and how inflammatory it is. And it's only after you become addicted to nicotine can you tolerate smoking. Now, the World Cancer Report, this was about 4 years ago, found that 18% of human cancers, over 2 million a year, were related to infection, not counting these kinds of inflammatory agents. And we've just finished the draft, actually, the proofs of the next report, which I'm one of many other authors. And this is a gross underestimate of the amount of cancer caused by the combination of inflammation and infection. Now, I mentioned obesity, and these are two seminal papers that were published 10 years ago in Journal of Clinical Investigation. And what these authors found is that in fat, there's high levels or high concentrations, frequency of macrophages, and these macrophages are activated and form pro-inflammatory loops with preadipocytes and adipocytes. So obesity is a chronic inflammatory disease, increasing the risk of cardiovascular disease, neurological disease, and cancer. And the macrophages also produce free radicals that can be either oxygen-based or nitrogen-based free radicals that can damage DNA, and if that repair is not repaired by base excision repair, you get mutations. But free radicals also damage proteins, including proteins that are important in DNA repair and in apoptosis. There's epigenetic effects of free radicals in terms of increasing DNA methylation, and also increases lipid peroxidation, produce these highly reactive electrophiles that damage DNA and again are repaired by base excision repair, and the lipid peroxidation pathway through COX-2 and COX-1 leads to cell proliferation, and certainly is important in a variety of cancers. Keep doing that. All right, so this is Jane and Aaron who asked the question about inflammation, and in this case, colon cancer. We've done the same thing with lung cancer. And what they found was that a combination of primarily pro or anti-inflammatory cytokines in the non-tumor or adjacent area as well as in the cancer together are associated with an increased risk of in this case, colon cancer. And in the adjacent non-tumorous area, high levels of the anti-inflammatory IL-10 are found, and that's also associated with increased nodal metastases in lung cancer and colon cancer. But in the tumor, these pro-inflammatory cytokines are at high levels, including IL-23, which enhances the proliferation of Th17 cells and produces high levels of IL-17, which frequently has a pro-tumor progression effect. So what happens if you combine, in this case, the inflammatory risk score with miR-21 expression? Is it a better prognostic classifier than either alone? That's the hypothesis. And indeed, that's the case. So these are Kaplan-Meier plots again, 100% survival, 0% survival. This is a 5-year point. High inflammatory risk score, primarily pro-inflammatory cytokines, poor prognosis, same patients, same prognosis, high levels of miR-21, poor prognosis. But if both are low, quite good prognosis. If either one is high, intermediate prognosis. And if both are high, very poor prognosis. It's very important to take these kinds of results to statistical analysis, particularly multivariate analysis, in which you're asking the question, is each covariant independent of the other? As you can see here, these are hazard ratios. The inflammatory risk score is independent of miR-21 expression, and they're both independent of tumor stage. So what's the principle here? Well, each have some value, and you get curves that primarily look like this, and you get a more robust prognostic classifier. But the rationale for saying that this is more robust is that each one of these, whether it's non-coding RNAs or coding RNAs as a biomarker, misclassify some patients. But they misclassify different patients. Now, that's putting two mechanistic and statistically independent biomarkers together. What happens if you put a third with these two? Is that better than two? The answer is yes. What happens if you put a fourth biomarker that's mechanistically and statistically independent? Is that better than three? We don't know. We're studying that right now. Is it only true in this case in colon? No, it's true in esophageal cancer. It's true in both adenocarcinoma and squamous cell carcinoma of the esophagus. And it's true in lung cancer, which I'll tell you about now. And then Carlo Croce's group has done the study in breast adenocarcinoma. These three came from our group. So this is a paper that was published about 2 months ago, and the hypothesis is the combination of protein coding genes are mechanistically related to lung cancer, and non-coding miR-21 is a better prognostic classifier than either alone. So let's look at these data in two different cohorts. One is a Japanese cohort in which it's primarily stage 1A, that's less than 3 centimeters, no evidence of metastases. So the 4-gene signature, which is increased XPO1, increased BRCA1, increased HIF1 alpha, and a decrease in a tumor suppressor, DLC1. So if those are the combination are high, then you get poor prognosis. If miR-21 is high, poor prognosis. If you put the two together, poor prognosis. So what happens in a second cohort, this is a US-Norway cohort, which is primarily 1B, that's greater than 3 centimeters. So you can see the prognosis is even worse in these primarily stage 1B patients. And the 4-gene signature, poor prognosis. miR-21, poor prognosis. And then if they're both low, that. If you're either one is high, that. And the two together, the worst prognosis. And again, multivariate analysis was done on these cases. We went to the literature and we found 5 other cohorts that had the same 4-gene signature that hadn't been recognized, but they, once we analyzed those, we found indeed that the 4-gene signature, protein coding signature, was associated with poor prognosis. And since we wrote the paper, 4 new cohorts of stage 1 adenocarcinoma have come up with essentially quite similar or the same results. And so we're writing a meta-analysis paper on this and some other aspects of lung cancer. And if you look at hazard ratios of all these studies, And these are relapse-free survival and cancer-specific survival. Those give you much more confidence when you're doing these kinds of studies than just overall survival. Although I must say in lung cancer, overall survival is a pretty good measure because once a person gets lung cancer, they die quite quickly, usually within a few months, as Terry and I know. So stage 1 lung cancer cases, after curative surgery, in quotes, 30% will reoccur and die of the disease. In stage 1A, which is a little better prognosis than 1B, the big difference is the tumor is a little bit smaller here and here, but in both cases, surgery is what is utilized. But we know that some of these people are at low risk of developing recurrence, and some are at high risk. So how can we identify those individuals? Because the high-risk individuals might benefit from adjuvant chemotherapy, and certainly would be a better population for doing the trials. So prognostic biomarkers, particularly those that are mechanistic, and use of gene expression is one way of going. Now, you heard in the previous talk about the National Lung Screening Trial, which has been replicated. And in fact, there is a 20% reduced mortality in their initial study. And most of these cancers that were actually cancers were stage 1A cancers. And secondly, the sensitivity of their assay was quite, the spiral CT is quite good, but the specificity is not so good. In fact, only 1 out of 10 of those tumors proved to be cancer. And as you heard, there are ways of trying to improve that. Well, one approach that we're taking, and we are Breed Ryan in the lab and Maya Holzner, is to look at pro-inflammatory cytokines. And so we've done some studies, prospective studies and prognostic studies of inflammatory cytokines in serum, and have found that IL-8 and CRP are associated with increased risk of lung cancer. And at the time of diagnosis, one finds, and this is as much as 5 years prior to diagnosis of cancer, and about the time of diagnosis, you see an increase in IL-6 in both Afro-Americans and European Americans. And in Afro-Americans, you see interleukin-1 beta and IL-10 increasing. In prognosis, you see a variety of different cytokines that are increased in the lung cancer cases, and some are more related to European Americans and some toward Afro-Americans or toward both. And we're currently writing up a paper now that shows even stage 1 lung cancer, IL-6 and IL-8 are fairly good predictors of who's going to have recurrence. So one strategy is to, and we have the serum samples, or we will soon have the serum samples from the study with the proper controls to ask the question to test this hypothesis. Another approach is unbiased metabolomics, discover biomarkers that are associated with risk, diagnosis, and prognosis and therapeutic outcome. And the use of urine as a biofluid goes back quite some time to Hippocrates, who tasted urine diagnosis of disease. Physicians at that time were rather more bold than we are today. And this is just an interesting quote. This is an interesting, at least to me, interesting painting that goes back several centuries. So this is a picture of Maya, who's working with Frank Gonzalez as a collaborator. Maya's a fellow in the laboratory. This is a strategy that we've used for identifying metabolites in cancer patients versus controls to be associated with cancer prognosis. And this is some of her data looking at 4 different metabolites, 3 which have been defined. This is the mass aspect of this unknown metabolite. 2 of these are found in tumors, and these are Kaplan-Meier plots. So 100% survival, 0% survival, 5-year. If high in all 5 of them, quite poor prognosis. And this is statistical analysis here. So in addition to serum, we have urine samples from the National Lung Screening Trial to ask the question whether or not metabolomics will help identify those who are at increased risk versus those who might not be. And lastly, I mentioned earlier that miR-21 has a lot of mechanistic data that we and others have gathered up to bolster its import in both diagnosis and prognosis biomarkers, but also as a target to therapy. So gene amplification, miR-21 is an area that's frequently amplified. It can be decreased by DNA silencing, and loss of that leads to higher levels. We've shown that the EGFR and the KRAS pathway lead to an activation of miR-21. David Baltimore and his colleagues have shown that IL-6 and interferon activate STAT3, the transcription factor that activates the miR-21. And with Nicole Simon and Jim Mitchell, we published a couple of years ago that different kinds of genotoxic stress can lead to an increase in miR-21. So what's downstream from miR-21? As I mentioned earlier, microRNAs have many targets. So those of you who do laboratory studies and you're using antisense, and I described some of those, you're very concerned about off-target effects. So you do everything possible to prevent any off-target effects, looking at multiple siRNAs and so on and so forth. But microRNAs evolved to have many, if you want, off-target effects. They affect the expression of many protein-coding genes. Not just one, but many. And these are some of the genes that miR-21 targets. And I selected these because they've been validated in laboratory studies. And secondly, they're related to cancer in one way or another. And this is just a partial list. And then the most recent, which I mentioned earlier, is that miR-21 can act as a ligand. So is miR-21 an oncogene? Well, in animal models, the answer is yes. Both Slack's lab and Olson's lab has shown, in this case is lymphoma, in this case is lung cancer. And the Slack lab has shown oncogene addiction. So what is oncogene addiction? So in an animal model, you overexpress a oncogene, you get a tumor, and then you turn it off, and the tumor melts away. That's called oncogene addiction. And is there oncogene addiction for miR-21? The answer is yes. So is miR-21 the only oncogene? And you have to be careful about cellular context. The oncogene that's a microRNA, no. miR-155, you get similar results, including the oncogene addiction. So the next question, is it a therapeutic target? So anti-miRs are being used in the clinic today. The first report was not for cancer, but for hepatitis C virus. So there was a New England Journal report that was published about 5 months ago, showing that an anti-miR that knocked down microRNA 1, 2, 3 in the liver is associated with a decrease in hepatitis C replication. And this microRNA actually enhances the hepatitis C replication. And it had been previously shown in monkeys that this was effective, and they staged, and now there's evidence in humans. A microRNA that's a tumor suppressor microRNA is in the clinic in stage 1 hepatocellular carcinoma and also lung metastases to the liver. And that study should be reported in about 2 months from now. Are there any studies of anti-MIR21 that are in the clinic yet? The answer is not quite yet. But there's a company called Regis that is about ready to, maybe it's already started a trial of using an anti-MIR against MIR21, which you might imagine is a pretty attractive target for some of the reasons I've talked about. So let's just briefly summarize this. P53 isoforms are physiological regulators of replicated cellular senescence, both in cells and culture and all of us in this room. That P53 isoforms are deregulated during carcinogenesis and in tumor-associated T cells. And we're studying the hypothesis now. I just showed you some preliminary data that P53 modulates the balance between symmetric and asymmetric cell division. The serum inflammatory cytokines are associated with lung cancer risk, diagnosis, and prognosis, including stage 1 lung cancer. That MIR21 is a mechanistic and an oncogenic biomarker of many types of cancer. And the combination of protein coding gene and microRNA gene expression is a better prognostic classifier in these cancer types than either alone. And then lastly, gene expression and metabolomic analysis may improve the diagnosis of early stage lung cancer by spiral CT. That's the hypothesis that we're testing, and we'll see what the results will be. And these are my coworkers on the left who have been involved in these studies. A number of them have gone to other laboratories, established their own laboratories, and our collaborators are listed here that I learned a great deal from all of these people. And I thank them, and I thank you.
So are you able to alter MIR21 by regulating exosome activity?
That's a very interesting question. We haven't done those experiments at all. Carlo Croce has. And so he has some evidence that different kinds of experiments. One is to knock down MIR21 in the tumor cells and then isolate in the exosomes in which have a deficiency of MIR21, and they don't activate the toll receptor. So those are the kinds of experiments that have been done. Exosomes can be measured in serum and plasma. There are a lot of kits that you can spend money on that don't work. So don't do that. You have to go through a 3 centrifugation process, and it's pretty laborious, but that does work.
Okay, well, go get some rest, Kurt.
Thank you for your patience.
Thank you for your lecture.